Great, it started.
Okay, so hi and welcome back to the 2023 PhysiCell Virtual Workshop and Hackathon. This is session 6, where we're going to be continuing building the sample model we have over the last 2 sessions. And today we're going to continue by adding inflammation, a basic form of T-cell attack. And the fun thing is we're going to actually add some mutations that help these tumor cells avoid the immune attack. And then an idea to model a therapy that can resensitize these cells to immune, basically by immune regulation here. And so, like before, we're going to assume the same structure for our projects, like PhysiCell and PhysiCell Studio alongside each other, and that I have just a symbolic link to simplify my structure. And what we're going to do is we're going to resume one of the models from yesterday. And so I'm already in my PhysiCell directory. And as you can see, I was working on a different project. So I need to go back and bring in the project I was working on yesterday with the guys. So I'm going to do, supposing I had unzipped it into my user projects. So I'm going to be looking for project 5 version 3. So remember, another way you can do this, right, is to do make list user projects. And it lists everything in that directory. I think we need to modify our makefile to not show the zip files. Will probably be a future enhancement to that makefile rule. But yeah, we want to load that project. So if you just downloaded it, first thing you need to do is make unpack, and then the name of the project. Proj equals session 5 version 3. And in this case, I'm expanding over the top, and then make and then load that project. Now sample session. And now everything's populated, and then make once more to compile it. And the special trick that I learned yesterday, and what you guys were trying to teach me yesterday, and I finally learned, is I have to go here now and open that file, that template.xml. So go here into config and open template. And here we are, right? We have the microenvironment with oxygen, drug, and debris. We have cancer cells and macrophages. And we have a variety of rules. I decided to get rid of the hypoxic adaptations for now and just keep it a little bit simpler. So I have the pressure and oxygen driving birth and death. And then I have the drug that is driving death. And I have dead cells releasing debris. And so what we want to do now is to continue this into a model that starts getting some inflammation, and then add in T cells that respond to that inflammation. So our approach here for adding inflammation is that we're basically just going to add a diffusible inflammatory factor. Also want to go in and kind of restore the boundary condition. Remember that we had played with the boundary condition just to kind of see the impact of our rules. Now I'm going to go back to a more uniform boundary condition of 5% oxygen or physioxic conditions on all 4 boundaries. And notice that these are all checked to on. So I've now added that here. I want to make sure I've got the chat window open. Okay, so we've got this. The next thing that I want to do now is actually, here's our strategy for adding inflammation. We're going to add an inflammatory factor, and I'd like to have an approximately 100 micron length scale and a slow decay and removal. And so I'll give it 1,000 is my diffusion coefficient, so that's 10 to the 3, divided by 0.1 makes this 10 to the 4, square root of that is 10 to the 2. So this has the length scale that I'm looking for, 100 micron length scale. Then I'm going to make a second macrophage type called M1 macrophages. They're going to be identical to my original macrophages, except that they also secrete a pro-inflammatory factor, the thing that we just added to our simulation. And this is going to be a persistent change, right? Rather than just saying that in some circumstances secrete pro-inflammatory factor, I'm going to say in some circumstances, I'm going to turn into a type that secretes pro-inflammatory factor to make that M1 cell type a little bit more persistent rather than a transient dynamic. And I think John is going to look at this nuance a little more in some of his sample projects, some of his warm-up models. So then the third thing I'm going to do is say, I want my naive macrophages to turn into M1 macrophages in some circumstances. And the way I'm going to do it says that when they're patrolling around, if they bump up against a dead cell, I'm going to use that right now as my promoting signal that will make them want to turn into a pro-inflammatory macrophage. So when they're in contact with dead cells, they're going to increase their rate of transforming into an M1 macrophage. And to decide how long to take it, I thought, well, it takes time, right? When you get a signal, there's actually some processing lag in a cell. It takes time to say, take a signal, go all the way down into the nucleus as a transcription factor, turn on a new gene, make some more protein, RNA, make some more protein, and drive a behavioral change, right? So there's some kind of time lag in that signaling cascade. So I'm going to make that transition rate on the order of 1 hour. So 1/60 is about 0.017 minutes inverse. So that's what I'm going to use as my choice of parameter value over here. So let's go ahead and start building it out now. So I'm going to go here into my microenvironment first. I'm going to make a new diffusible substrate. I'm going to double click it to change its name to pro-inflammatory signal or factor. And I can set my diffusion coefficient to the value we talked about before, 1,000 with a decay rate of 0.1, no initial condition, nothing in the environment before we got here, and nothing on the boundaries. So far, so good. The next thing I'm going to do is I'm going to go and make this M1 macrophage type. So I'm going to go here into my cell types. I'm going to select the macrophage type. And I'm going to go ahead and just copy it. And then I'll double click this thing and call it M1 macrophage. And I'm going to leave him pretty much exactly the same as everybody else, except I'm going to go into secretion and say that this M1 macrophage should be consuming oxygen like everybody else, no drug, should be consuming debris because that's part of what macrophages do. And it should also be secreting a pro-inflammatory factor. So I'm going to set my secretion rate to a high enough value that it kind of picks up a local value around here. I usually just default to 10 on a secretion rate just to kind of get a good secretion. And don't forget to set a non-zero target. So I'm going to just say that I'm trying to secrete fast enough to get that factor to be close to 1 if I'm near a cell that's secreting. So I pick the target of 1. Say I want my local concentration to be close to 1 wherever I'm secreting, and a secretion rate 10 to kind of force it to that value a little bit faster. Okay. And then I'm going to go and add a new rule for my macrophages. So go to the Rules tab. And remember, we need to say that when I'm in contact with dead cells, I want to force a fast transition to an M1 macrophage. And so I'm going to add a rule, say for the cell type macrophage, my signal is going to be contact with a dead cell, and that is going to increase my rate of transforming to an M1 macrophage. Half max 0.5, hill power 10 is probably reasonable because remember, we want this to be kind of 1 or more cells should be enough to spur this transformation. Although actually you could pick intermediate values here, and you could say that maybe I need to be in contact with several dead cells before I want to transform. And that's something you could actually explore. My saturated value is saying that once I'm in contact, I'm going to go with that 0.017 rate that we talked about. And I can plot my response curve and see how when I'm in contact with no cells, low transformation rate. When I'm in contact with 1 or more dead cells, I've got this higher transformation rate. So we click on Add Rule, and we save this thing. Make sure that I'm not applying it to dead cells. That's kind of important. And let's go ahead and, now the thing is, when I load a model, it doesn't load the initial conditions in the editor. So I'm just going to go ahead and recreate that. Then I'm going to make a circular region of cancer cells. Maybe let's do more than normal. Let's do 250 micron radius or even 300. A little bit bigger of an initial tumor this time. And then I'm going to go ahead and say, let's add in some naive macrophages. And let's randomly put them in this annulus. Let's say between 350 and 400 microns, we'll place 100 of these guys. And let's go ahead and save that. And let's give it a run. Let's see if these new rules now increase transformation to M1 macrophages and make sure that they're secreting pro-inflammatory factor. So I can go here and do the initial plot, we already see, if I do a legend, I see cancer, macrophages, and when macrophages are yellow. Let's change the coloring here so we can see a little bit better. So I'm going to change the contour plot to that lovely blue. Let's do it this way. Now I can kind of see the cancer cells against it a little bit easier in the immune cells. So we can see that they've already come into contact with dead cells and they are transforming into M1 macrophages. And now let's see here. Do I have drug? Yeah, I have drug on the corners, but it's decayed away pretty quick. Do I have debris? Let's turn off the cells. Yeah, I have debris being secreted by the dead cells. And do I have pro-inflammatory factor? Yeah, definitely. And it's you can see it's localized around the secreting cells, these M1 macrophages. So far, so good. We've got some inflammation now, at least some inflammatory signaling. And we've got these cells transforming into macrophages that are continuing to consume, but now they're secreting the pro-inflammatory factors. So we can start kicking off the rest of our immune model. Does this make sense before I go on? Okay, cool. Thanks, Alzadeh. Appreciate that. And yeah, we can. There are ways to control where to put these clusters. That's a great question. Actually, let's go to immune conditions, and just for the fun of it, I'm going to make another little tiny micrometastasis, maybe right up over here. So my center for this guy should be, let's say, at 300, 300. So x0, 300, y0, 300. Inner radius of 0, outer radius, let's just go very small. Let's say 50, maybe 75 microns. So now we have a little bloop of tumor cells over here. So actually, we can go ahead and run that. So here we have, remember, that initial bolus of drug is causing some die-off on the cells. And these guys struck pay dirt. They're already in the tumor. So they've found some dead cells to turn them into M1 macrophages, and they're starting to consume the debris. And everything goes on as it does. So we'll go ahead and keep that example. That just kind of show we can place cells in different locations. Okay, so let's go ahead. Well, we ran it and visualized it, and that's lovely. Let's start adding in some T cells, some cytotoxic T cells or effector T cells. And so let's talk strategy before we do it. We're going to add effector T cells, and they're going to be exactly the same as macrophages, except instead of eating debris, they're not going to eat debris. They're not going to phagocytose dead cells, and they're not going to uptake prolactin. They're not going to attack. They're not going to eat. They're not going to consume debris. They are not going to secrete pro-inflammatory factor, but they are going to follow the pro-inflammatory factor by chemotaxis. So they're going to be drawn to the inflammatory signal from the macrophages. And they're going to attack tumor cells, which will cause damage in the cancer cells. And then remember, we need one more hypothesis that says, what do we do with that damage? And so we're going to say that the damage is going to increase apoptosis or death in the cancer cells. So once we add both of these things, then we have a good T cell model. So, so let's go ahead and do just that. So we're going to go here to the cell types. I'm going to just grab the regular macrophage. I'm going to copy it. And I'm going to call this thing an effector T cell. And let's go through the properties now. Oh, and okay, so we've got this. We've got the mechanics, motility, all this. Now for motility, right, I want them to be chemotaxing not towards debris, but towards the pro-inflammatory factor. So I changed that here in the motility tab. And for secretion, let's see what this guy should be doing. He is consuming oxygen. I think that makes sense. He is not consuming drug. He is now not consuming debris, because that's not the job of a T cell. And now I think we should give him an uptake rate for the pro-inflammatory factor. And the reason why is because if you're sensing something, you're not doing it by magic, but by a receptor, which means that if you're sensing some molecule it implies that you are binding to that molecule with a sensor, with a receptor, and bringing it in. And so that means that any rule that says that you respond to some signaling factor probably implies that you should have a non-zero uptake rate of that corresponding factor. So that's a good rule of thumb, I think, when you build a model. Okay, so we have that. Now we need to go to the interactions, make this thing act like a T cell and not like a macrophage. And so we've got this. We're going to go to the interactions. We have to make sure that he is not phagocytosing dead cells. So set that rate to 0. Make sure I'm not phagocytosing anything else either, but we never set rates to be non-zero, so they should be fine. But he should be attacking cancer cells. And I'm just going to pick a rate of 0.1. This says that if I've been in contact with the cell in the order of 1/0.1 or 10 minutes, I should give it an attack. So, attacking fairly frequently. And this is basically a parameter value you're going to have to experiment with to really see what it does. This is, I think, an area where the immune models for us are much newer than other models. So I think we can use a handful of good default parameter values for these things. But I'm going to set 0.1 just based upon playing with these for a long time. And let's see here, do I need anything else? Oh yeah, that's right. I need to go and add the rule that says, right now I have T cells attacking tumor cells and causing damage, but that damage needs to cause death. And so I'm going to go here into the rules tab, and I'm going to say that for cancer cells, damage increases apoptosis. And let's see here. I'm going to pick 5. 5, just saying I need to have accumulated, 5 units of damage, basically 5 minutes of attack time. And, I'm going to just pick hill power 4, just to have something kind of gradual. And I'm going to pick the same 5 times 10 to the negative 3 as my response. So you have the same response for now as the other thing. So let's see what this rule looks like. So this says that as I accumulate damage, my rate of death goes up until I saturate towards some higher value. Let's go ahead and add that rule and go ahead and save. And I think we're ready to give this a go. Oh, no, we're not. This is not very helpful if we actually don't put any T cells in our environment, right? So we better go to the initial mutations tab. And now I've strayed from my script a little bit. So I'm going to put the T cells kind of in the same mixed band. Actually, I'm going to make them a little bit farther out. So I'll put them in a band centered at 0, 0, between a radius of, let's say, 400 and 450. And I'll put 100 of these things in. Now, there is some overlap, but the mechanics will work it out. I'm not going to worry about it. So let's go ahead and save that. And let's give it a go. So hopefully we are seeing some T cells now being drawn to the inflammation by the inflammatory factor and then attacking cancer cells. So first of all, here's our initial condition. Let's look at our legend. So in this legend, cancer cells are gray, macrophages are red, pro-inflammatory M1 macrophages are yellow, and effector cells are green. Apologize for the palette. Hopefully you guys can see these, especially when we start plotting. So we definitely see, again, the macrophages are polarizing the M1 macrophages. And if I change to, say, debris, I can see that dead cells are releasing debris that are drawing things in. You can see the sharp edges of the debris field. That's where the macrophages are consuming that debris. So you can kind of see that they are cleaning up the environment a bit. They're very good garbage men. And then if I look at pro-inflammatory factor, I can see that these M1 macrophages are definitely secreting and drawing it in. And let's see what happens. So we've gone 14 hours. And we can definitely see that these effector cells seem to be making some headway here. Right, the tumor looks like it's being nibbled away a bit and shrinking. So one thing we could do is we could actually go over here and click full list, and we could plot the damage. And so let's kind of rewind here and see how the cells that have been attacked are accumulating damage until they die. And the macrophages are just really, really effective at phagocytosing the dead cells, so you don't see these apoptotic cells lingering for very long. But now let's look at the population plot. I'm so glad that you reminded me of that, Randy. You see how the cancer cell population is steadily decreasing from that effector attack. So that is really cool. So we have built a fairly effective and robust immune response over here. So Anna, thank you for reconnecting. The slides will be available on the repository for the workshop. So if you go to github.com, PhysiCell Training Workshop 2023 or WS 2023, we'll go to that main page, click on Agenda Materials, and go to the full link over here, and you can scroll down and look for session 5. So here's day 1 activities. My scroll is not very fast. Here's day 2 activities, and we're in session 6 right now. Oh, okay, I need to fix and add the link to the slides. I apologize for that. So one way I can do that. Session 6, here's my slide deck. I'm just going to copy my link, and let's just edit this thing live, because real people just edit the code live and their GitHub's live, right? Let's see, let's go to agenda. I'm going to edit this file and find session 6. I really apologize. I thought I already had done this, but apparently not. Paste that in. And now the slides are available and linked in. Yeah, here we go. Right there. Click this link. Okay, so I'm glad you brought that up, because apparently I forgot to post the link. Let's see here. And let's go to the end of our simulation. We do have a little bit of crowding of some cancer cells managed to escape over here, that little cluster, which is a bit interesting. We can actually see that the population rebounded a little bit and is coming back under control. We can kind of play out the movie to see what the dynamics are that brought that about. But more or less, there just wasn't enough death over here to really get a steady presence of pro-inflammatory macrophages to draw in the T cells. So it really is kind of escaped attention, just because there's not enough death to bring in and focus those macrophages, and they're really not going deeper into the tissue. So that's, immune infiltration is a big deal, right? And that drives a lot of good and bad behavior of actual immune responses. And so that's, I think, a topic worth exploring a bit more. Okay, so we have an effector attack, but that's no fun. Let's start adding some cancer resistance. Questioning, yeah.
Yeah, sorry to interrupt. Would you mind closing your Outlook reminders just so the screen is?
Oh, yes.
Thanks.
Outlook is being a pest. I really appreciate you bringing that up. I've gotten so good at ignoring those that I just didn't even see it. Thank you. Which doesn't make it a particularly effective reminder, does it? Okay, so we've got that. Yeah, we're doing way fine on time. So let's go ahead and keep adding fun things to the code and see what we can do. So we added our effector T cells, and we plotted and visualized. And so this is version 2, although I have now a slightly different initial condition than you guys have had. I think it's, let's do the next thing. I'm going to add basically some immune evasion now. And so one thing we know that cancer cells can do is over time, they can become a little bit less sensitive to immune cells. They can actually evade immune predation. And there are a number of theories on why they may do it, but we're going to model that just as a generic, say that they become less immunogenic. That is, that they are less interesting, let's say, to the immune cells. Remember, an effective attack requires 2 things: a high attack rate of that specific type by the attacker cell, in this case, the T cell, and also immunogenicity to the attacker by the victim cell, I guess, in this case. So you need both those things to be good to get a successful attack. And so what we are going to do is we are going to add a rule that changes the immunogenicity of the tumor cells. And so our strategy, this is a pretty short strategy slide, but I'm going to add one rule for now, just for an easy thing, that says that when a tumor cell is in contact with an effector cell, that it is trying to reduce its immunogenicity. So it's kind of actively trying to modulate that, and it decreases immunogenicity. Now, I can say that there are other ways to model this that actually would be a little bit better. And in a future version of PhysiCell Studio, if you go to cell types and go to interactions, there's going to be another field here soon that will let you control the immunogenicities of that cell type. And when that is there, then we would be able to say maybe tumor cells mutate to another cancer cell type that has low immunogenicity. And then you could just have that as a built-in parameter. So that would be a way to model it more persistently. But for now, we're just going to do it as a transient reduction in immunogenicity whenever they're in contact with a T cell, they try to do this. And so so that's the strategy I would have liked to have done, but we will do this. And actually, we're just going to do it by adding a rule. So go over here to the Rules tab, and I'm going to select Cancer Cell. The signal is contact with an effector T cell. And that is going to decrease my immunogenicity to effector T cells. And see, and let's suppose if I'm in contact with one cell, which means I'm going to put my half max halfway between 0 and 1. So 1/2 with a very steep response curve of 10 power. So, and I'm going to say that reduces my immunogenicity from a base value of 1 to 1/10 of that. So here's my response curve. When I'm not in contact with any T cells, I'm immunogenic to cancer cells, to T cells. And once I come in contact with one or more cells, I am much less immunogenic. So let's go ahead and add that rule and save it. And let's go ahead and run this thing. So run simulation. So there's a question in the chat saying, in the plot, if I want macrophages to return to baseline, can I apply changes in the GUI? I don't think I understand that question. Are you trying to go back to initial conditions? Are you trying to change the appearance of the plot? And maybe we'll just circle back to that question. So here we have the initial tumor, and you can see that initial drug response is causing some death here. And you can already see some of the macrophages polarizing. And now, they are drawing in the effector cells, but you see a lot more cells left behind. So kind of punching their way in there and attacking, but really not having a lot of effect. So let's change our plot to visualize that further. Let's look at, I don't think we can look at the immunogenicity, unfortunately, right now as a field that we can plot. So that we may need to add into our standardized outputs, but I can plot the damage. And if they're being attacked less, there should be a lot less damage accumulated. So let's see how that looks. Yeah, we can see some damage, but it's never, it's definitely accumulating much, much slower. Remember that used to get up around 15 or 20 on this scale. Now it's getting up around 1/3 of that. So let's go back to the regular view. You can see now that just this little tiny tweak of a rule on the cancer cells is allowing them to much more effectively evade that immune response. So cancer fights back in this model. Now, this is just a very basic version. There are models we could build, I think, if we had with the fuller API right now and later on with the GUI that allows us to do this in an even more nuanced way. But that is a start. So in the future, I would have said, let's make it more controllable by actually having a separate cell type called resistant cancer cells or something like that. And the biggest difference would be that they have a lower immunogenicity for all those cells. And then you would add a transformation rate from the cancer cells, the mutated cancer cells. And then the other thing is you would have to, right now, we would have to basically copy all the rules we use for cancer cells. We probably want to apply them to the mutated cancer cells. So right now, there's no notion of inheriting rules in the simulation. So we would probably want to copy them. We may want to think about ways to copy masses of rules together, but for now we can do that. Okay, but we have that. Now let's do an anti-immune therapy. So the cells became anti-immune, so we're going to do anti-immune, or maybe resensitization. So we're going to add a new therapy, some kind of theorized monoclonal antibody or something, something with moderate diffusion and low decay, and that the effect of that rule is that it's going to supercharge our T cells. It's going to say that they're going to try to attack more. So basically, I could try a rule that either brings that immunogenicity back up, or I could do a therapy that makes the T cells better at attacking, even if they're not very immunogenic. And so we're going to take the easy approach here and do that. And so the way we accomplish this first, right, is we're going to go into the microenvironment tab, and then click on a new variable. I'm going to call this, I'm not very imaginative this morning, so I'm going to just call it therapy. And maybe your marketing research people can come up with a cool name like cancer kill TM. There we go. Okay, our marketing department decided to get to work and called it cancer kill TM. There's a TM on there, so that means it's a registered trademark, so no one can steal that. All right, and I'm going to give it a diffusion coefficient of 1000, a very low decay rate of 0.001, and let's just set it up to be 1 everywhere on the boundary, and make sure to apply it to these boundaries here. Okay, so now I have cancer kill TM on all the boundaries. And then the last thing I need to do is add a rule for that therapy to have an effect. So I'm going to go over here to the rules tab, and I'm going to say that in effector T cells, cancer kill TM increases my attack rate on T cells. And I can just go up by a factor of, so I don't think I want it 0.1. I think I want this actually to be 1. Right, because, so let's go ahead and try that. Oh, and let's do a more reasonable half-max of Hill power 4. So it's a little bit more gradual of a response curve. Okay, so I'm going to add that rule. We've got a good set of rules here. Let's go ahead and save it. And let's give it a go. Let's see what it does. It's funny because in my picture, I think I have the saturation value of 1. So I just need to make this slide deck consistent with that. Okay, so let's see what we got here. Are the cancer cells doing T cells doing a better job of attacking the cancer cells? Yeah, I mean, they're tunneling their way in, and they seem to be chomping quite a bit more. And we could plot the damage to see if they're accumulating more damage now. So look at the horizontal scale, right? The scale is going up closer to 1520 like before. So we kind of restored the overall function of this. We should be seeing some debris from all this cell killing, so that's a good sign. And we still have the pro-inflammatory factor. We have the obnoxiously named therapeutic. So I think this is looking pretty good. Yeah, you can definitely see, I mean, visually, you can see the big difference on the behavior here. So that works there. So someone asked, can you transition to a different cell type, or is a change of behavior following a signal? So we could always add another cell type and have a transition to that cell type and have a bunch of different parameter values for that new cell type. That's definitely possible, just like the differentiation starter model we did this morning. So that is always a possibility. In fact, let's go ahead and do this. Let's have one more cell type called, let's delete that. Let's copy cancer cells. and call it mutant cancer. And let's just change its behavior a little bit. Let's suppose that mutant cancer cells have motility turned on. They want to do oxygen chemotaxis, so going up oxygen gradients. And let's suppose that these cells, actually we could make them, let's assume that these are really smart cancer cells. And let's have them do chemotaxis away from the pro-inflammatory factor. And then we're going to add a transition rate. So we're going to say that cancer cells transform into mutant cancer cells at some kind of low background rate. So that's something that we can definitely have them do. Now, the thing is that the mutant cancer cells have not inherited all the rules from the cancer cells. So we may need to copy some of these rules, for example. And so what may be the way to do this is to go into, oh, that's not a text editor. I want to find my terminal shell. I think I have it on another monitor and I can't find it. Too many windows strikes again. I will find it. There it is. Okay, Zoom was hiding it from me. Okay, so I'm going to open this directory and I'm actually going to open the CSV file. And I'm going to overwrite the stupid Macintosh defaults. So I can open this up in a real text editor. That's amazing. I can identify myself as the developer, but apparently I can't convince Macintosh of that. I'm going to try to force it. Yep, take that, Macintosh. Okay, so I'm going to grab all my roles from cancer. And I'm just going to copy them in. And let's see, our new type here, right, is, let's make sure we get the name right, because I'm doing this manually now, right? My cell type is mutant cancer. So I'm just going to take all of these and write the word mutant in front. So I've got all these rules now that are applied to mutant cancer cells. So I took all my cancer cell rules and I just copied them. And this is a bit of an experiment on the fly, so please bear with me. But I'm going to go over here into the Rules tab, and I'm going to import the rules that we just overwrote by hand. Oh, yay, they all showed up. So that's good news. Okay, but let's see how our test went. Remember, we just tested this therapeutic. And we see that the cancer cells are doing a much better job. I'm sorry, the effector T cells are doing a much better job now of attacking these tumor cells because they've been kind of reinvigorated effectively. But now let's try with the new rules and see how it goes. Now we have cancer cells at a low background rate mutating into mutant cancer cells. And there's nothing different about them at all, except they're a little bit more motile. Well, actually, there are a few things different, right? They're more motile. They move away from the inflammatory factors, and we have not actually programmed the T cells to recognize the mutant cells. So they should be basically very well ignored. So let's run this thing. Uh-oh, I didn't like something. So I'm trying to scroll up through the console output. Could not find stem cell. That's hilarious because there are no stem cells in this model. I don't know what its malfunction is here. Yeah, this is what we get for going off script here. Just try it again. These are some debug things I've added in the past.
Paul, when you imported the rules,
Uh-huh.
I don't think it should be necessary, but did you also save from?
Oh,
I don't think it's the problem, but
well, let's try that. That's a really good idea. So these are fairly normal. I have a fairly verbose output right now because I'm trying to debug something. I don't see anything that's wrong with this. The next thing I'm going to try to do is run it from the console instead. I don't know. Drug increases. Well, I think we're gonna have to come back to this one. For now, I'm going to comment out some of these rules. Really, the one I want the most is the negative feedback from pressure so they don't overcrowd everything. So I'm going to go here and I'm going to import the rules again. Oh, that's interesting. Just ignore them altogether for that. Cool. Okay, let's see what happens. We're running. Okay, well, I just commented out most of those rules and we can worry about that later. So now, if I do my legend, I have this new mutant cell type emerging. And I probably should have done a much lower mutation rate because we have quite a few of them becoming these cells. Is it just me or these M1 macrophages doing something different than we had them doing before? It looks like they're phagocytosing something. Oh, well. Well, we'll sort that out later. So the interesting thing is we did get a rule that they were transforming into these mutant cells, but we don't have the rule that says oxygen increases cycling in those mutant cells, so they basically die off over time. Not by immune predation, but just because they're not particularly proliferative. So we would need to further develop that to do it. On the other hand, it looks like there's a whole lot of death. And so the macrophages did a whole lot of eating. That's why they're seeing them getting really, really big. And then they digest the materials they just ate and they just get smaller again. Okay, so we winged it a little bit and we added some things. But I think more importantly here, we were able to basically model an anti-immunity therapy, or I guess immune resensitizing therapy that would supercharge the T cells and get them back on the attack and manage the tumor much better. So this is actually where I wanted to be on the immune therapy sample model. Interestingly enough, we've finished early again. Last year when we taught this, it took us to about Thursday to build this model. So I guess I'm happy with that. And we even had a little time to wing it and improvise as we were going. So I'm pretty happy with that too. But please take this not as a demonstration of what you should do in your models, but more as a demonstration of some of the different capabilities that can be connected together to build more complex models. You can add things like differentiation and transformation. And you can add things like secretion, consumption of chemical factors. And that you can make rules that say how the cells respond to contact with other cells, or to diffusible factors, or to gradients. And those are kind of like the language that you want to combine to build these broader hypothesis-driven models. So I'm happy to look at questions now. Alzadeh asks, if during infection, immune cells recruit cells to infected sites, and all immune cell populations die over the course of the infection, so in the end, they return to baseline. Oh, that's what you're looking at. Yeah, you can definitely do that. What you can do, actually, and we do in more sophisticated immune models, is you can have the immune cells proliferate and die as well. And so one great rule to add, for example, is that you can have, let's say, T cells, for example, proliferate in the presence of pro-inflammatory factor, and you can have the M1 macrophages primarily secreting pro-inflammatory factor when they're in contact with cell debris. And so what might happen is if the attack finishes, then there are no more cells to kill, which means the macrophages stop secreting pro-inflammatory factor, which means that the temporarily increased proliferation by the T cells is removed. Now, on the other hand, you have a death rate of those immune cells, of those T cells, then that's going to come to dominate, and eventually that population will go back down again. And that's just the simplest possible arc that you can add. If you add anti-inflammatory factors as well, you can accelerate that response. So you can have, for example, that M1 macrophages, when they're not in contact with cell debris, might want to transform into M2 macrophages and start secreting an anti-inflammatory factor. And you could have proliferation go up as a function of inflammatory factor, and you could have apoptosis go up as a function of anti-inflammatory factor. And so that would kind of compete and start cycling down the T cell population. That's just, again, one possible feedback that you could add, but we really need to be driven by the biology on this. So you can always think about how pro and anti-inflammatory factors could modulate differentiation, either into a T cell or naive T cell. It could not modulate rates of proliferation, it could modulate rates of death, it could modulate rates of migration. Right? So all these things that are things that you could think about secreting. Of course, it can modulate how fast they perform attacks or their damage rate. So we have a lot of things that we can manipulate based upon whatever our hypothesis might be to try to get that population to return to the baseline, for example. And I think that's also a great notion that many great cancer models are actually started by modeling the underlying tissue dynamics first. Let's say you're trying to come up with reasonable parameters for your tumor angiogenesis model. Well, make a fake wound and no tumor cells in it, and see if you can repopulate that tissue. And if you can, you might have some good parameter values for your stem cells and anything else you might have. Let's say your blood vessels, for example. So hopefully that answers some of that question. But thank you so much for rephrasing it. Any other questions? So go ahead and stop the share here. And let's go and stop recording too, please. So thanks very much for joining us. This is the end of